Let's see if we can almost get done with our lecture for this chapter for today. We'll get, I think, pretty much all the way down until we get to actually some of the biochemical processes for our carbohydrates and lipids and proteins. We were talking about the stomach and the composition of the stomach mucosa, the gastric pits, and now we're into the gastric glands. And the gastric glands indeed will have mucus cells, part of the protective mechanism for the stuff that's bubbling up from the gastric glands. And then we will encounter as we go on down, and this is actually the order in which we encounter them typically, we'll encounter the big fat parietal cells, which are responsible for acid production, hydrochloric acid, as well as intrinsic factor. Then we come down further on down, and we'll see these smaller, more pigmented or stain uptaking cells called the chief cells that you should also know as zymogenic cells, because when we talked about the parathyroid gland, we also had another cell called chief cells. So don't forget that there's more than one area that we see the word chief cells. So these are better known as zymogenic cells. They produce the protein digesting enzyme called pepsin. And they produce it in an inactive form, and it's the acid environment that it bubbles up into that activates that protein digesting enzyme. So that acid pH is necessary for activation. Then as we go further on down, you can find the enteroendocrine. So entero is gut, and endocrine, which means that this is a hormone that we're producing, right? Enteroendocrine. This is a gut hormone and produced by that enteroendocrine G cell, and that's nice that they called it an enteroendocrine G cell because the hormone that they produce is gastrin. So that's really nice that they actually kind of added that to the descriptor for that. Gastrin is a hormone that obviously is going to circulate in the bloodstream that is designed to actually stimulate the production of more gastric juices. And remember, gastric juices is hydrochloric acid and pepsin, and obviously water associated with that. But that's what when we say gastric juices, I'm talking about hydrochloric acid and activated pepsin. So this cell you cannot identify unless it's a special stain, and we don't typically do those special stains for these cells. But you can identify the parietal cell, which is big and fat and pale staining, and then the chief cell, which is much smaller and takes up a darker stain as you move on down the gastric glands. Let's talk about the various digestive processes that actually occur in the stomach. pH 2 starts protein breakdown because pepsin is activated by hydrochloric acid. There are 3 phases of gastric juice production. And that's what's gastric secretion, gastric juice production. And it's interesting that we actually secrete anywhere from 2 to 3 liters of gastric juice a day. So gastric juice production is going to have the cephalic phase, the gastric phase, and the intestinal phase. 2 are stimulatory, 1 is inhibitory. The cephalic phase occurs before food enters the stomach and is what I equate to anticipation. So let's talk about bacon. I have already started your cephalic phase. The name, just the word bacon, the thought of bacon, the smell of bacon, the sound of bacon, the visual side of bacon are all going to create the cephalic phase of gastric juice production. So this is anticipation. Sight, sound, smell, and anything else, touch, whatever. All of those are considered part of that cephalic phase. So we're going to get some gastric juices being released into the lumen of our stomach is what this is telling you before food has ever arrived there. Then we're going to hit the gastric phase, which is going to cause the bulk of our gastric juice production. And that is due to gastrin, which is a hormone that is going to cause the release of hydrochloric acid from our parietal cells. Now, this is an interesting process. When food enters your stomach, what does it do to the size of your stomach? It distends it. So a baroreceptor is going to be kind of part of this process. So stomach distension, so food distends stomach. And this is a neural mechanism that's going to stimulate gastric juice production. Plus, we'll get a dilution of the stomach pH. So if I have a container of acetic acid, which is, vinegar, right? And I pour in a bunch of water, is it the same pH as it was before I poured in a bunch of water? The pH goes down or up? Up. So that's another mechanism here. So pH change here, which is a chemoreceptor response, is also going to cause gastric juice production. So dilution of stomach pH, again, chemoreceptor response, because a neural response with distension is a pressure receptor or mechanoreceptor or baroreceptor, whichever one you want. Well, then also all of these, distension of stomach and dilution of stomach pH, will all produce gastrin release, which is a hormone, which then means we will get an increase in gastric juice production. Another thing that's important that also produces gastrin release is caffeine. I always spell caffeine wrong. So if you drink a cup of coffee, that will actually stimulate gastrin release, and that will put more stomach juices or gastric juices in your stomach. And if you have a stomach ulcer, is that a good thing? No. And so that's one of the things that often they'll say if you're trying to take care of a peptic ulcer or stomach ulcer is, stop your caffeine consumption. No coffee, even no tea, because it also does the same thing. No Red Bulls or whatever your energy drinks are. And those are all going to stimulate more hydrochloric acid production and irritate the stomach. So those are the main causes of gastrin release. Well, now, once we're done with the meal, we need to inhibit gastrin release. And so that's the next phase. The intestinal phase opposes the cephalic and gastric phases. And we do this because contents are now reaching the small intestine, and that's why we also call it an intestinal reflex, because this is going to be protective of the small intestine, more specifically the duodenum. We want to inhibit gastrin release. If food is entering the small intestine, we don't need to be putting a whole bunch more stomach acid into the stomach, because now we're actually going into the small intestine for further digestion and absorption. So let's inhibit this gastrin release. And This is also by the same mechanism that stimulated gastrin production in the stomach. So distension of the small intestine. So as we start squirting in the acidic chyme, that's a distension mechanism or a baroreceptor. And then plus pH change. Because in this area, it's constructed very different than the way we actually have the lining of the stomach constructed, because it's more for other digestion and absorption. And so the pH change, which is a drop, is also going to be stimulating the inhibition of gastrin release, because acidic chyme is entering into that area, and we don't want that acidic chyme breaking down the duodenum or the lining of the duodenum and creating a duodenal ulcer. And so we need to inhibit gastrin release for that. So distension, baroreceptor, pH change, chemoreceptor are all going to inhibit gastrin release. So that means we're not going to make so much stomach acid or stomach juices. The other thing that actually happens too when food starts entering into the duodenum, we will get a release of two other hormones, secretin and CCK. That stands for cholecystokinin, and that was mentioned earlier in the notes on this chapter. And so it's so much easier to actually point out secretin and CCK. Secretin is a hormone that is actually released due to pH. A drop in the pH stimulates secretin production from the duodenum, and CCK is going to be released when we have lipids entering into the duodenum. Now, I want you to think about a meal. It's going to have water in it, it's going to have protein in it, it's going to have carbohydrates in it, it's going to have fats in it. Put it in a jar and shake it up a lot. Stop shaking it for a moment, and will it layer out any? Yeah. It will layer out, and what's at the top? Because that's what I want to talk about first. Fat. That's right. Fat floats, correct? And it still does. Fat always floats. I was always one that was envious of some people that didn't have problems treading water, because I could tell the difference between a muscular person and one that wasn't so muscular. The treading was a lot more work. Okay. So it always was. You guys remember doing the dead man float? Did you ever have to learn how to do the dead man float? I was always vertical in the water. I never could get horizontal. I was just like, something's wrong with my feet. I always thought when I was a kid. I said, yeah, I got bony feet evidently. So, but of these two hormones, secretin and cholecystokinin, which one's secreted first? Secretin. And so that when I ask you this, that obviously means there must be one that's secreted before the other. And then CCK is released because lipids get in later. Both of these, both secretin and CCK, this is what stimulates their release. Drop in pH and presence of lipids. That stimulates their release. And both of these are designed to decrease gastrin hormone release and therefore decrease gastric juices. And doesn't it make sense? As you are emptying your stomach, you don't need any more of those gastric juices released. So we have stimulatory phase, cephalic, and then gastric, and then we have an inhibitory phase, which is the intestinal, because stomach's emptying. So it's really a pretty good mechanism to take care of things for you. Gastric motility. Stomach typically empties about 4 hours after a meal. And so the three muscle layers our circular, our longitudinal, and our oblique muscle layers. I'm just churn the food into chyme, which is acidic. And then our pyloric sphincter opens up for to let's see, just 3 cc's of acidic chyme into the duodenum at a time. Let's say per peristalsis. So the stomach will churn, then it will stop churning, and it will have a nice peristaltic wave, which remember is squeezing the tube of toothpaste, towards the pyloric sphincter. It will open just enough to allow 3 cc's of acidic chyme into that area. Then that peristaltic wave will cease, that sphincter will close, and so we have now that acid being deposited into that area, and even just that 3 cc's is enough to actually cause some distension. It was empty before, now it's being distended, and so then we actually have the initiation of secretin release in order to actually start to slow down the acid production for the stomach. High-fat meals delay gastric emptying. So 4 hours after a normal meal, but if you have a high-fat meal, it might be 6 hours, 7 hours, or 8. It just depends upon the individual. So that's the story about the stomach. Now let's go into the small intestine, which is where our acidic chyme is going. And this is important. We did a little bit of carbohydrate digestion with salivary amylase. We're in the stomach with acid being poured on food, as well as a protein digesting enzyme pepsin, so we're getting a big protein digestion occurring in the stomach, and plus with the hydrochloric acid, some of our carbohydrate digestion is occurring, but not as much as when we're actually getting into the small intestine and the rest of this digestive tract here. But we have not started to digest really lipids to any significant extent, and so now we're depositing those into the small intestine. And so it's important that actually you realize most of digestion, physical, which is going to be segmentation, and chemical digestion is going to occur in our small intestine, followed by absorption. In the cadaver, the small intestine is about 20 feet long, and it's only half as long in live people. Do you have any idea why we have a length discrepancy between cadaveric small intestine and living? It's made out of smooth muscle and smooth muscle tone. Absolutely. And so we've lost our smooth muscle tone in the cadaver, and so it's kind of stretched out in the cadaver. The first division of the small intestine is the duodenum, and it's approximately, in us, only 10 inches long. Then we come to the jejunum, and in that area, it's actually only about 8 feet long. And then the rest of the small intestine is the ileum, and that's approximately 12 feet long. And these are obviously cadaveric numbers, and you know, with a little fudge factor here and there in those. The ileum terminates at the cecum via the ileocecal valve. And the small intestine, because we are going to do a lot more digestion and major absorption here, we have to change the surface of the lumen, of the border of the lumen of the small intestine or of the mucosa. And so that's why we have here modification of tunics designed to increase absorptive surface area. Plica is fold in Latin. So plica circularis is circular folds. And these are visible to the naked eye. And so right here, plica. Is what that little leader line is actually pointing to. So you can take a look in, put a scope in, and you can easily see these folds increasing surface area. Then off of the plica circularis are villi. Now villi aren't visible in a way that you can actually recognize to the naked eye, but they give the surface what feels like and looks like a velvety appearance. In the middle of these projections, because the villus is a projection, are capillaries, blood capillaries, and lacteals. A lacteal is a lymphatic capillary. And lacteals are named that way for lactation. And what is the coloration of products of lactation? And that's why they're called lacteals, because these lymphatic capillaries, instead of being clear and almost difficult to see, they're actually white and easy to see. And of the material that you typically think of as white, what do you think of as far as foodstuffs would be mostly colored white? It's actually fat. Is it fatty material typically white when you look at it? Well, actually in our specimens, they're yellow by the time you guys take a look at it. That's part of age and preservation, obviously, process. So these are actual projections, and it says finger-like, and I want you to remember finger-like on the microscopic level. And so what we're referring to here, I prefer this picture down here. This is a villus. Do you guys see that? So those projections are villi. And with this circular fold cartoon up above, they're actually referring to those little guys as villi, but those aren't things that you would actually see unless you put some sort of dissecting microscope or handheld magnifier to actually be able to see those. So those are the villi, also increasing surface area, and then microvilli. Well, remember, those are surface modifications of our cell membrane, and so our notes say projections on the columnar cells, and obviously increase surface area significantly. And what's interesting, they actually have surface enzymes. Remember back to first semester when we talked about making up plasma membranes? Remember, it's a phospholipid bilayer, and what is always embedded in it? We talked about proteins. Remember, we have transmembrane proteins that create pores for us. But remember, we also have peripheral proteins. And that's what I'm talking about with surface enzymes, because enzymes are structurally proteins. And so we even have digestion actually occurring with those surface enzymes on those microvilli. So histology of the small intestine, columnar absorptive cells with the microvilli obviously, and goblet cells are seen there. And as we scroll on down the page to the actual histology view, you can, I think, even with this lower power, you can easily pick out goblet cells from that view, yes? The ones that look a little more like this guy here. He's got the little, what appears to be a hole, so goblet cells have that mucus in them that typically get removed whenever we fix them and take a look at them. Another term that you will sometimes see with microvilli is right down here. It's brush border. Can you understand why they call it brush border? Because it does look like a little bit of the bristles of a broom, so to speak, or a hairbrush, that type of stuff. We're now down to a very important organ, the liver, which is our largest internal organ. It is an accessory structure of our digestive system, and it is responsible for producing bile because we need to start working on lipid digestion, and we do so with a fat emulsifier. You guys remember, has anybody here ever made mayonnaise, homemade mayonnaise? What else? I have too. So you take a whole bunch of oil and you put it in a jar, and then you add an egg and shake it up, or you can take your egg and beat your egg with a whisk and then slowly drizzle in oil and keep drizzling it and keep beating it vigorously and keep beating it and keep beating it. And it's predominantly all oil with some egg in it. The egg, what we're doing is, because we're whisking it and beating it, we're breaking it up. As you know, when you take fat on top of, or oil on top of water, you can run a knife through it and it will want to come right back together. Well, because of an emulsifying agent, which is what the egg actually helps to provide for you, as you try and make that line in it, that emulsifying agent will keep a separation between the fat. And so as you break it up, make it smaller and smaller, because of the emulsifying agent, those little fat lake becomes millions of little fat molecules, or not molecules, but globules. And the smaller you make them, that actually creates when you work them all together, larger surface area. And because of that larger surface area that you've created, then the digestive enzymes can actually break them down to the monomers that we need for actual absorption back into the digestive system or the blood vessels of the system. And so that's what bile does. Fat's an emulsifier and makes the big fat pool all these teeny tiny little globules that digestive enzymes or lipases can actually work on. Anatomically, our liver has 4 lobes. One of them is really large, and it's the right lobe of the liver. So on the right side of your body is where the bulk of your liver is located. You do have a left lobe, and you have 2 other lobes that I'm not going to mention, but they're all smaller. The right lobe of the liver is indeed the largest, and that's where you're going to find the gallbladder hanging off of. And this little teeny tiny picture down there actually shows you the large right lobe of the liver with the gallbladder peeking out from the anterior margin of our liver. We do have a mesenteric cord that actually helps suspend the liver from the diaphragm and the ventral body wall. It's called the falciform ligament. And within the falciform ligament, in the inferior aspect of it, is the round ligament, also sometimes referred to in the literature as ligamentum teres. And it is a remnant of your umbilical vein. And so remember the ligamentum arteriosum, the connection between the pulmonary trunk and the aorta. It actually scleroses or scars on down. That's what this is too. This is our umbilical vein. We no longer need it, and so it's now scar tissue. And that's in the inferior aspect of the falciform ligament. The liver, when we take a look at it under the microscope, has this interesting lobule form. So structural and functional unit of the liver, so realize this is kind of a microscopic view of things. And we'll call it a microscopic cylinder. It's going to be composed of hepatocytes, and they are going to be radiating around a central vein like spokes in a wheel, bicycle wheel. And so let's take a look at the cartoon and then a little bit of the histology view of that situation. Okay. Central vein in the center of each lobule will then drain the blood into hepatic veins on the back side of the liver into the inferior vena cava. At the perimeter of our lobule are these things called triads, and there's 6 of them. Triad means 3 structures and 6 of them arranged around a lobule. And in the triad, we will have an hepatic arteriole, which will contain fresh blood, which means O2 rich. And hepatic portal venule will also be right there next to it, containing used blood coming from the intestines. So used means CO2 rich, correct? O2 poor. But if it's coming from the intestines, what kind of venous blood is it? Just O2 poor, CO2 rich? What else does it have inside of it that was absorbed from the capillaries? Into the capillaries, I mean. Our foodstuffs, correct? And so it will be nutrient rich. And it will be toxin rich. Because we ingest toxins. Alcohol is a toxin, acetaminophen is a toxin, ibuprofen is a toxin, and we are going to send all of this stuff to the liver for processing and detoxification. So all of this blood goes to the liver. The liver is a major processing plant. Also part of that triad is going to be a bile duct for bile transport, and this is going to actually go in the opposite direction. Flow-wise compared to the arteriole and the venule. And let's take a look at the cartoon right down below it. So this is a CV, central vein, CV, central vein. And here is our venule over here. Here is our arteriole next to it. And here is our bile duct. And so that's all part of our triad right there. And take a look at the direction of the arrows, which is meant to indicate the direction of blood flow through these hepatocytes. And if you'll notice, the arterial blood is actually joining up with the venous blood, and they are both flowing to the central vein. So what this is meant to show you is the liver never gets as much O2 content in the blood as the rest of our organs. So our O2 content that the liver lives with is always going to be less than 104 partial pressure. Do you guys see this when we mix arterial blood with venous blood in our liver? Our hepatocytes can live at a lower PO2 than the rest of our organ systems can. So we are mixing arterial blood and venous blood, but it's still enough for the liver cells to get the oxygen that they need, to get the nutrients that they need, and then to actually process the toxins and to actually repackage the nutrients to actually allow this blood, this nutrient-rich blood, with proper processing of the nutrients into forms that are now reasonable to send to the heart, to send to our systemic organs. But first, if it's the IVC, where does this blood go to? So our liver is going to actually dump this blood that it's processed via hepatic veins into the inferior vena cava. Where does the blood go to next?
Right atrium.
Goes into the right atrium, because this is actually used blood, correct? And then it goes into the left, right ventricle. I think I must be very tired today. And then from from the right ventricle, it goes to the lungs for oxygenation, correct? And so this is nutrient-rich blood going to the lungs for oxygenation. And then now it's nutrient-rich, oxygen-rich blood that's going now to the left atrium and then the left ventricle then to send that blood to the rest of your body. So it's an interesting way, but it's still a way that actually works. So the liver processes the foodstuffs that's going to be delivered to your body first before the blood gets actually oxygenated. It is just the way it is. So our notes here talk about the hepatic portal system. It is designed to take O2 poor, CO2 rich, nutrient rich, again, doesn't hurt to say it over, toxin rich, blood to liver for processing before allowing it into our venous blood. Which is going to take it straight to the heart, and then to the lungs, and now we'll send everything good stuffs to the body. This is kind of in the wrong spot, this intestinal veins, and I'll talk about it now. And then we'll go back to the biliary system after we take a quick look at this histology view. When you take a look at this, at first sometimes you might think lymphatic. Can you guys think lymphatic when you take a look at this? One of the things that it's reasonable because you see a bunch of nuclei, but this central vein and then these areas off to the perimeter will end up being typically visible and obvious in a histology slide for you guys to actually ever take a look at, project it for you. But CV, and hopefully you can see the radiating pattern of the hepatocytes out into the perimeter. So this doesn't look like a lymphoid nodule or follicle at all to me. And so if you're in lymphoid tissue, the bulk of it has nodules or follicles, except for obviously the thymus and the spleen. And so, but spleen and thymus look very different also from this. And so the liver, though, sometimes you first look at it and you might think lymphoid tissue, but then think, no, we don't see any nodules and it doesn't have any of that thymic appearance or any of that to it. The nice thing about the histology view from above, it shows you the vein here, huge, like most veins are. The artery or arterial next to it is small and muscular, and I'll erase that from the view so you can see that. And the other thing that kind of gives it away is that biliary duct. It's got cuboidal cells in it, and that's a really obvious structure. It actually is quite cellular in its appearance. It hasn't, all of those structures do have lumens associated with them. Intestinal vein-wise, for us and even for your cat, the hepatic portal vein is short when you compare it to other veins. In your cat, it's full of yellow latex, correct? The hepatic system. And we have one venous leg coming in, creating our hepatic portal vein, and another venous leg coming in, and they're both mesenterics. There's one called the superior mesenteric, and that is going to be taking this blood that it is getting from the small intestine, from all of the small intestine, some of the large intestine, and then also the inferior mesenterics are actually taking venous blood to the liver from most of the large intestine. There's a little bit more detail than that, but I think superior mesenteric small intestine, inferior mesenteric large intestine. And then they all empty into the hepatic portal vein, which is emptying into the liver for processing. Again, this is notes are kind of out of sync here. Composition of bile. Let's go down and just talk about the gallbladder, and then we'll talk about the composition of bile. Gallbladder is defined as a thin-walled green sac embedded in the liver, and it is a bag that stores bile overflow. So it's a storage or reservoir for bile overflow, so to speak. So bile is not produced in the gallbladder. Bile is produced in the liver, and then the liver is going to dribble bile into the biliary system, and then the biliary system is going to fill up and overflow into the gallbladder. So it's designed to actually be a storage reservoir for bile, for its actual overflow. Its walls have smooth muscle. And will contract when a certain hormone is released. And we just mentioned it, and it's up above in your notes, which of the two hormones, or three, because gastrin's a hormone, secretin's a hormone, and cholecystokinin is a hormone, which one, based upon the name even, is going to be the one that's going to CCK. That's right. So CCK stems GB, gallbladder contraction. And that's the nice thing too, even about the name cholecystokinin, because I don't know if you've ever heard of what the name is for a removal of a gallbladder, but it's called a cholecystectomy. So cholecystokinin and a cholecystectomy, and so CCK actually stimulates the contraction of the gallbladder to release bile. Now bile itself, all by itself, is kind of yellow-green, and it's also alkaline. And actually that makes sense, doesn't it? It's going to go into the duodenum. What do we know it's there for? It's there to help with emulsifying fats, which are just also part of that fatty kind that now is, not fatty, but acidic kind that is actually entering into the duodenum and fatty. And so it's nice to have it be a bit alkaline to help neutralize the pH, correct? So that's really good. And the two things that are in bile that I want you to know about are bile salts and bilirubin. And bilirubin is actually what helps to give bile its color. Bile salts are made from cholesterol. So it's interesting, bile salts are the actual emulsifiers. Of bile. So bile salts are the emulsifier of bile, and so they'll help us to take this big fat wad and create millions of little teeny tiny fat globules here, and that is done to increase surface area for proper enzymatic digestion. Interesting thing about bile salts, almost all of them are recycled in the ileum. And what I mean by that is they're reabsorbed. So we use bile salts for emulsification, breaking up into teeny tiny little fat globules. Then we pour enzymes onto them, and then the bile salts are still active in floating down the intestinal tract and reabsorbed in the ileum and recycled and used over again. So that's what I mean by recycled. We actually absorb them in the ileum and use them over again. Bilirubin is indeed a yellow waste product that is from the breakdown of the unrecyclable aspect of hemoglobin and heme. So this is not recyclable. And bilirubin is what our gut bacteria use to turn our stool brown. So let's talk about why we can actually have a cholecystectomy, which is the removal of our gallbladder. Why do people get their gallbladder removed for the most part? Gallstones. And you want to know what's really interesting, the most common material that gallstones are made out of is actually cholesterol. So like a pearl in an oyster, you get a little bit of too much cholesterol precipitating out, and then you start getting layer after layer after layer of cholesterol around that and actually creating your gallstones. So you can have one, you can have many, you can have what I call grit. There's gallstones come in all different forms, but that's their origination is actually cholesterol deposition. And bile itself is highly concentrated cholesterol, so it actually makes sense that there's actually that cholesterol issue with this. If you get a stone, the problem with the stone is it can obstruct the gallbladder when it's contracting, and that's when you become symptomatic. You can have stones, and as long as there's no obstruction of the gallbladder when it's trying to contract, you will not know that they're present, and so there's no reason to mess with something that actually isn't bothering you. But if you get a stone, the gallbladder is a bag, and you guys, many of you have already done your GI dissection eval. Remember, it tapers on down and narrows into, what's the duct that leaves it? Cystic duct. Okay, so we have that cystic duct, and it's tapering down, narrowing. The cystic duct will tee into another duct called the common hepatic duct, and once it tees into the common hepatic duct, it's called the common bile duct as it goes and empties into the duodenum. The reason they call the area above the TN where the cystic duct is common hepatic is because we have bile ducts coming from the right lobe of the liver and bile ducts coming from the left lobe of the liver, and they converge together and create that common hepatic duct. So that's why we have that name. Now, the liver is producing bile 24/7, and the bile is in the biliary system. You saw that the bile was going in the opposite direction that the blood flow was, which is towards the biliary tree that I've just talked about. So it's dripping into the right bile duct and the left bile duct into the common hepatic duct, and then it's dripping all the way down into the common bile duct to the hepatopancreatic ampulla. You guys remember all these things from your GI eval. Now, it fills that tube all up, and when it gets overfull, then bile goes into the cystic duct into the gallbladder. You guys follow that pattern? Because the gallbladder is actually a storage site for the overflow of bile, and then when we have a high-fat meal, we will have contraction of that gallbladder, which will deliver a bolus. You guys remember what bolus is? Large volume. It will deliver a bolus of bile to the fat and allow now for us to mix it up vigorously to emulsify that fat. And so that's what the gallbladder is for. The gallbladder is designed to give you a bolus of bile to the high-fat meal. Now, if you have a cholecystectomy, a removal of the gallbladder, that means you can no longer give a bolus of bile to a high-fat meal. You guys follow that? That's one reason why you can remove it. And where would you cut? If you were to cut the gallbladder out, where do you cut in the biliary tree? What is it that you're supposed to properly tie off and then cut in order to remove it? The cystic duct. Because if you cut the common hepatic duct or the common bile duct, you're in big doo-doo, correct? Because what you have done, if you cut the common hepatic or common bile duct, you have no longer can bile leave, correct? If bile can't leave, it builds up and it builds up in the liver, and from the liver it will build up where? In the rest of your body. And it will create what color in you? Yellow, and you'll be jaundice. And so jaundice is obviously inappropriate bile in the system from various reasons. In the newborn, it's because their liver isn't mature enough to process bile properly and to create the products that we actually need to get rid of. So we can't actually break down and get rid of our bilirubin because the liver isn't doing its job yet because it's immature. So that's why jaundice occurs in the newborn sometimes. But in other people, when we have hepatitis, liver inflammation, the liver cannot process bile and create bile properly anymore and get rid of it, and so it overflows into your bloodstream, backs up into your bloodstream, and that's where that situation actually occurs. But if you cut everything properly, cut the cystic duct, you still have bile that's able to go into the rest of the duct system, into the duodenum properly. Now, what kind of muscle is going to be part of the duct system and the gallbladder itself? Smooth muscle. What's so cool about smooth muscle that we like it for our bladder? You can stretch it. So you have to understand the duct system is also all smooth muscle. And so the liver, remember, is going drip, 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 drip into this tree that no longer has the overflow reservoir. And so what do you suppose over time will happen to the little straws, so to speak, of the duct system over time? They start to expand. And so that's the other nice thing. And that's why typically after a cholecystectomy or removal of your gallbladder, they'll tell you to limit your fat intake, because if you eat too much fat without the bolus that occurs, you end up with what's called fatty diarrhea, because it overloads your gut and your gut can't handle all that fat getting further on down the digestive tract. It causes that type of process. But over time, so people will actually cut back on their fat in their diets, but then over time they'll start sneaking more and more fat in. And that's because humans are sneaky that way. They like to eat the stuff that they like. And they'll be able to tolerate that because over time, the biliary tree is no longer the skinny tree it was. It's the little more rounder tree that time actually gives us. So there'll be a lot of people who say, oh, I can eat all kinds of fat in my food, and no matter what I ingest. And I go, yeah, I know why. Time has given you many favors. So this picture here is also showing us the pancreas, which is always associated with the duodenum. You guys realize that the pancreas, if you have to lift up the cat's stomach, right, in order to find the pancreas, correct? Same thing with us. So our stomach is a C, and the back of the C is to our left, and our duodenum is another C, with the back of its C going to our right. And the head of the pancreas lives in the C of the duodenum, and the body runs across, and then the tail actually likes to touch and tickle the spleen. And so that's how the pancreas actually is in us also. And it's flat. It's a flat organ. It's a little bit more, there's a little bit more glandularity to it in the human, but it has a lot of similarities that you find with the cat. This is showing you here, this is the duodenum here. The stomach has been removed, and that's why the picture shows you the head of the pancreas, the body of the pancreas, and the tail reaching off to the spleen. The pancreas is retroperitoneal. The duodenum is retroperitoneal. So that's one of the things to actually remember. Retroperitoneal. The common bile duct is going into the duodenum posterior to its surface, and also even going to go into the pancreas posteriorly, and then through the head of the pancreas and body of the or meat of the pancreas, not body, and will end at the hepatopancreatic ampulla, because the pancreatic duct, we actually have several, but let's just concentrate on the main pancreatic duct, goes through the meat of the pancreas and meets up with the common bile duct at the hepatopancreatic ampulla. And then that sphincter is going to be associated with the hepatopancreatic ampulla, and that will relax when secretin or cholecystokinin is actually released for us. So that's a little bit of that story for that picture there. Regulation of biorelease, well, we actually already figured that one out. Trigger, fatty chyme enters the duodenum and causes CCK release. And then also, and this is to stem GB contraction. And also CCK release also stimulates pancreatic juice release. And so when we say juice, we mean there's many different things in this liquid. Going on down to the histology of the pancreas, we see two views. This one is very familiar, correct? Pancreatic islets. And then the one adjacent to it off to the left, that is the bulk of the pancreas. It's called acinar tissue, A-C-N-A-R, acinar tissue, because the units making them up are acini. And so we have a lot of cuboidal cells that are actually around creating this kind of structure with a center area that you can't actually see too well, but ultimately has a little lumen where the juices actually can go. As far as the functions, we've already discussed the endocrine function of the pancreas. The pancreatic islets secrete the hormones into the bloodstream. That's what endocrine is all about. Everybody remembers insulin as one of the hormones from the pancreatic islets. What's the other hormone from the pancreatic islets? Glucagon. Remember, insulin is designed to decrease blood sugar by allowing glucose entry into the cells, and glucagon is actually designed to increase blood sugar when our blood sugars actually get too low for us. So they both work on glucose levels to actually make sure we have a nice stable blood glucose level throughout the day and night. The pancreas is predominantly, though, as far as histology goes, an exocrine organ with all of that acinar tissue. And the pancreas will secrete its enzymes in an inactive form. And if we take a look at the cartoon up above, it actually tells you the basic enzymes that the pancreatic acini actually secrete. So we actually have amylase, which is to digest carbohydrates. We have lipase, which is now to digest our fats. We have proteases, which is to actually digest proteins. And we've already talked about a protease released by the stomach. And which one was that? Pepsin. That's right. Pepsin is a protease, but it's released by the stomach. So we have other proteases in the pancreas. And let me actually indicate one of the proteases that the pancreas releases, and that is called trypsin. Is a protease. So amylase is a carbohydrate. Let's put carbohydrates up here to remind us that that is the main carbohydrates that is actually being released. So amylase, lipase, proteases, and nucleases to digest DNA and RNA that it's going to encounter from our cells that it's breaking down. And these are all go into the pancreatic duct. And CCK, don't forget CCK, stems pancreatic juice release. The bulk of pancreatic juice, good old-fashioned H2O, but interestingly HCO3. Do you know what we want to call that ion? It's the bicarbonate ion. And what will that do to the pH if we add bicarbonate? because it's an alkaline. So it's designed to help neutralize also our acidic chyme. So pancreatic juice itself is alkaline. So alkaline pH, it's approximately 8, by the way. And this is to neutralize the acidic chyme in the duodenum. And because of this, we can keep pancreatic enzymes inactive until duodenum. Because we don't have goblet cells lining all of this, protecting us from the digestive effects of these enzymes. And so all of these enzymes are actually released in an inactive form, or created in an inactive form in the pancreas, get into the duct system, and then enter into the duodenum and become activated by pH change. So they're in an alkaline environment, and then when we put them into an area where we have acid, that acid chyme is also the activator for many of our pancreatic enzymes. So it keeps the pancreas from digesting itself. So now digestion in the small intestine, no significant fat digestion until now. We've been working on our carbohydrates and our proteins, even a little bit on the nucleic acids. So now we're in the small intestine, and segmentation is the mechanical prospect of mixing chyme, bile, and the enzymes to help them get physically digested and then mixed up with all of the enzymes that we've actually added in there. So segmentation is clamping down on one end of the small intestine, clamping down on the other end of the small intestine, and pretending the small intestine has teeth by its little muscles, chomping down on the contents and mixing things thoroughly, breaking them down physically as well as enzymatically. After we've had significant mixing, we will then absorb the stuff, and then peristalsis occurs after a good mixing plus absorption. For that area. And then the next area will do the same thing. Until no nutrients left. So segmentation, mix, absorb, and then peristalsis, move it on down, let that area see what it can do. Segmentation, mix, absorb, peristalsis, keep moving it on down until you get to the large intestine with no nutrients. And that's great because the large intestine's main function is the absorption of water and the elimination of feces, things that we cannot digest. Anatomically, the beginning of the large intestine is the cecum. Sac-like beginning of the large intestine. Is that in your right lower quadrant or your left lower quadrant? It's in your right lower quadrant. The appendix hangs off of the cecum. So that's where you find the appendix. Then we have four major divisions of the colon or large intestine after the cecum. We have the ascending colon that's going up right side to hepatic flexure. Then we have the transverse colon that is going to go from right to left to the splenic flexure. Then the descending colon, where foodstuffs or actually fecal materials going down left side until you get to the sigmoid, which is designed to bring to your colon and contents to the midline of the body. And then it will terminate in the rectum, which is a short section of the colon that's in the pelvis and will connect to the anus via the anal canal, which is the very last inch of the rectum, and we have two sphincters, the internal, which is smooth muscle, and the external, which is skeletal muscle, and the external is obviously surrounding the internal. And so we'll draw our ascending, our hepatic flexure, our transverse, our splenic flexure, our descending, going into the sigmoid, bringing everything to the midline, our anus and our anal canal and part of the rectum. Let's draw the picture to associate with this. Appendix hangs off of that, not quite like that. And then the ileum will actually tee in here at the ileocecal sphincter, I-L-E-U-M, C-E-C-U-M, and then rectum and anus. So the sphincters, internal anal sphincter, and then we'll have an external anal sphincter. The internal is smooth muscle, the external is skeletal muscle. One we have control over and the other we do not, correct? And so no matter what other parts tell us, as long as we have control of the outermost aspect, we're going to be able to actually keep things in the rectal vault itself. We'll talk about the defecation reflex next time. Let's talk for just a quick moment about tenae coli. Longitudinal muscle of the large intestine is divided into three bands, which will then cause the remaining colonic wall to actually bulge out into haustra. So lumen of the small intestine, lumen of the large intestine. We have a circular layer to muscularis externa on both, correct? Which is close to the luminal aspect. And then our muscularis externa also has a longitudinal layer, and I'm doing it like this because this is like a cross section. So if it was cross section, we cut across the longitudinal layer. Well, with the large intestine on humans, unlike the cats, we actually have all of those longitudinal muscle layers bunched up. And that's what the tenae coli are. And because we have them bunched up, the areas in between have less support, correct? In between the tenae. And so these areas have a tendency to be bulging outwards into those structures called haustra. So tenae coli right here, here, and here, creating the bulges called haustra. So bulging in between, and this is an area that has less muscular wall supporting it. And we'll talk about how that's important in diverticulosis next time. So we'll stop there. Sorry, just a little longer than I probably should.